When I say that I study cancer, most people think that I study cancer cells. While this is in part true, it misses a very important element: normal cells. Because normal cells within a tumor may be the key to understanding why certain patients with apparently similar tumors sometimes respond differently to the same therapy. Same tumor, same drug, but different response. My name is Marco Varrone, and I'm a PhD student in the Department of Computational Biology at the University of Lausanne. And in this video, I'm going to show you how our work zooms out to get the full picture of a tumor. Imagine a tumor sample as a fruit pie. Here, every fruit is a cell. For example, blueberries may be cancer cells and strawberries may be cells of the immune system. And the way both normal cells and cancer cells are arranged constitute what is known as a cellular niche. For example, the presence of immune cells may slow down the expansion of a tumor. Less than 20 years ago, the way to study these samples was through a technology called bulk RNA sequencing. This technology is equivalent to taking all the fruits, blending them, and try to understand which fruits compose the pie by looking at their color, their consistency, or the taste of the smoothie. Around 10 years ago, a new technology was invented called single-cell RNA sequencing. This allowed to separate each individual cell and characterize it. However, patients may have similar proportion of cells, but these cells may be arranged differently and compose different cellular niches. And finally, less than 5 years ago, this technology saw another revolution: the spatial revolution. This technology allows to characterize each cell while keeping information about its original location in the tissue. However, one last step is missing. How do you move from knowing the location of each cell to identifying cellular niches? That's my work. I developed a method called CellCharter. It finds cellular niches in tumor samples, even for samples containing millions of cells. It will still take a while before the special technologies reach the clinic. But when they'll do, they'll be able to divide patients based on their cellular niche and help finding the best therapy for each of them. And so a patient will come to the hospital and a test will tell him or her the type of cellular communities that are in its tumor. And a doctor will prescribe a drug specific to those communities to clear the tumor cells while preserving the healthy ones. Thanks for watching, subscribe to the channel, and see you soon.